Good evening. My name is Andrew Westby and I'm the Deputy Vice-Chancellor responsible for research and knowledge exchange at the University of Greenwich. It's an absolute pleasure to welcome you all here this evening. So, Jonne, Rodenburg, actually joined us in January 2018. And I've got pleasure to say I was on the interview panel. So it's partly my fault, But what it's proved to be has been a really, really good decision that we made on that day to appoint Jonne as a senior lecturer in agroecology. And he's made really, really good progress since he's been here. He was promoted to an associate professor in 2019 and to a full professor in 2022, which is fantastic progress. So before joining NRI, he was an agronomist at the Africa Rice Centre, where he conducted research on cropping systems across the continent. He was based in Malawi, Senegal, Tanzania and Côte d'Ivoire and worked in projects in those countries and also in the surrounding countries. He did his undergraduate and postgraduate studies at Wageningen University in the Netherlands and embarked on a BSc programme there in tropical land use in 1994. Gosh, it starts to show age, doesn't it, when you put years on these things. Specialising in agronomy and soil fertility for his MSc. His idea at the time was to do agricultural extension work in international development projects, particularly in Africa. And this was strengthened by a placement he did in Burkina Faso in 1997. However, a year later, he went to Indonesia to be involved in a research project at the World Agroforestry Centre. I think this kind of changed his life, and research was the way to go. And so there came the birth of the research career. So he did his PhD research again back at Wageningen University on the role of sorghum genotypes in the interaction with the parasitic weed striga, with field work at Ikrisat in Malawi. And this was one of his introduction to one of his main research themes of his career. So Jonne has two main lines of research, one in sustainable intensification of cereal cropping systems through agronomic innovations, and the other in ecology, biology and management of parasitic weeds. And this is, I think, more the parasitic weeds side of things that we're going to hear about tonight. So I'm not going to say any more because I don't want to spoil anything that's coming up. But, ladies and gentlemen, I introduce it to you, Professor Jonne Rodenburg.
Thank you very much, Andrew, for that nice introduction. And thank all of you for being here on a Friday evening. When I planned this date, I was actually only thinking about my family coming from the Netherlands, and I thought, well, if I would do it on a Friday evening, that would be nice, just before the weekend, but not thinking that this would not be the best day for all of you to be here now. So I'm really pleased that you are all here. Especially pleased that my collaborator from Kenya is here. He arrives today, Stephen Bruno, and of course my family from the Netherlands. So the title of my lecture sounds perhaps a bit intriguing. That was meant to be. I got a lot of comments and questions about it that I did not really want to go into at that time, but now is of course the time to reveal what it is all about. It is about the biology, ecology and management of parasitic weeds. You will not be completely satisfied now, but trust me, I will come to that later. But first, I have to do a bit of administration because Ben and a few others told me that it is good practice, actually a sort of a tradition, for new professors, if they do their inaugural lecture, to show themselves a photo of themselves in a football team, preferably in Africa. So I want to tick that box off. So this was in Benin in 2005. I was terrible at football, but we had fun. Now back to the topic. On the right-hand side of this slide, you see a word cloud that is drawn up from all my published work. Although half of it is indeed on agronomy and agronomic innovations for sustainable agriculture intensification, the other half is on the topic of this lecture. But the word cloud quite well presents what I will be talking about tonight. On the left-hand side, you see a sort of a table of content. I will talk about what parasitic weeds are, what the relation is with witches and vampires, so you will get your answer to that. How did I end up with them? Why did I study them? And did I find out how to get rid of them? And then I will present a brief conclusions and acknowledgements. So, the first question, what are parasitic weeds? This is a very simplistic and not so nice picture of a plant. You will all recognize that this is a plant because it is green. Yeah, so it has leaf chlorophyll, and that is the pigment that makes the plant green. It absorbs the red light part of the spectrum, of the light spectrum, for photosynthesis. There are, however, also plants that do not have chlorophyll, or less of it, or less functional, and those are the parasitic plants. You can categorize them in holoparasites, hemiparasites, and facultative parasites. I will first explain you about the holoparasites. Holoparasites, and you see that they are not green on this picture, they are red. They can also be yellowish or whitish, they vary in color. They depend on other plants for all of their nutrition. The hemiparasites, they are green, they depend on the host plant for part of their nutrition. So, holoparasites and hemiparasites, that is the big first distinction in categories. But I'm going to talk in this lecture only about hemiparasites, so forget already about the holoparasites. The hemiparasites, they are tightly linked to their host. And that is shown on this picture. What all parasitic plants have in common, holoparasites, hemiparasites, facultative parasites, root parasites, stem parasites, they all have a haustorium. And the haustorium is a specialized organ that only the parasitic plants have, and that is, enables them to connect to a host plant, to a host root or host stem, and to penetrate into the host tissue to make a connection, a vascular connection, in order to take up the nutrients, water and sugars from their host. It all starts with the soil seed bank. So the seeds reside in the soil. And when there is a host plant growing in their vicinity, they can sense it because of the root exudates and the compounds that are in the root exudates. If they sense the vicinity of a host plant through those compounds, then only then they can germinate. And an important class of those compounds are the plant hormones strigolactones, and I will come to that a little bit later again. So then they can attach to the host root through the haustorium, and then they have the below ground development. They will emerge above ground, they will flower, produce seeds, and the seeds are then returned to the seed bank, and it starts all over again. And they produce many, many seeds. I'm talking thousands of seeds per plant, millions of seeds per square meter. So that is also a very important component of this problem. When they attach to a host plant, they attract water, nutrients, and sugars from the host plant, and in return, they send plant hormones, proteins, all sorts of compounds, amino acids, etc. And that results in stunting, wilting, and chlorosis of the host plant. In fact, the host plant is severely affected by the parasitic plants. There's another category of parasitic plants, and those are the facultative parasites. The facultative parasites also start from a soil seed bank. Then they germinate and grow in a plant, just like any other plant. But when they sense the vicinity of a potential host plant, then they will try to attach to the roots of that host plant. And then, of course, they will have the same devastating effects on the host as the other parasites I was talking about. And in return, the parasite really benefits from this attachment. The growth of the facultative parasite is really boosted by it. It produces a lot more flowers, a lot more seed, when it is able to attach to a host. But this does not yet answer the question, what is a parasitic weed? A parasitic weed is when you find those parasitic plants in a crop. So that is simple. And when the density of the host plants is really high, then it becomes a real problem. Then the parasite becomes really successful, produces a lot of seed, and builds up a seed bank. And it becomes a big weed problem. On this picture, the above pictures, you see Striga hermontica in sorghum and millet crops in Africa, Côte d'Ivoire, Ethiopia. And on the below pictures, you see Striga asiatica in rice. Both of these parasitic plants, parasitic weeds, henceforward I will call them parasitic weeds, are widely distributed over Africa. And they have pretty colors, so they're quite nice plants. Another parasitic weed that I have worked on a lot is Rhamphicharpa fistulosa. Rhamphicharpa fistulosa is also widely distributed over Africa. The flowers are less shiny and colorful, but it is also a devastating parasitic weed. It is a facultative parasitic weed in rice, and here you can see pictures from a rice crop from different parts in Africa, where the parasitic weed is really hammering down the rice. Now, what is the relation with witches and vampires? From the title. So, the common name of Striga is witchweed. So that is a very short and simple answer to that question. Why are they called witchweeds? Because they have a long below ground phase during which they are invisible, but they are harmful to the plant already. So the plant is affected by it when it is attached, when a parasite attaches to the plant, and yet you cannot see it yet. And it has really a bewitching effect on the host plant. You have this stunting, chlorosis, etc. That is why it is called witchweed. The other parasitic weed, Rhamphicharpa fistulosa, the common name of that is rice vampireweed. And I was actually the name giver of that parasitic plant, because when I started doing my work on Rhamphicharpa in 2006, it was really not studied a lot. And we realized with my team that it didn't have a common name. So we gave it a common name, rice vampireweed, because it really clings on the host and then really drains it out. All of these parasitic weeds I'm talking about here have similar hosts, and what they really have in common is rain-fed rice. That is where all these parasitic weeds come together. Now I will show you some pictures of affected host plants, just to get you an idea of how devastating they really are. This picture shows 4 plants, 4 sorghum plants. But you may only see 2 or maybe 3. That is because these are the sorghum plants that are growing alone, and these are the sorghum plants that are growing with a parasite, host plus parasites. So the purple flowers is the parasite, and you really see that the host is completely vanished. Now this is just another variety of sorghum, so same effects. You hardly see the host anymore when it is infected by the parasite. This is rice infected by Rhamphicharpa fistulosa, the facultative parasitic weed. On the top picture you see the rice in a vegetative stage, and on the below picture in a generative stage, in the productive stage. And you see when the host is growing alone, it's doing quite well, but when it is infected by the parasite, it's the same thing as with Striga, very much diminished production biomass, etc. And what is unique about Rhamphicharpa, because it is a facultative parasite, it doesn't need a host. It can grow a perfectly normal plant without the host. But you see that the host is also clearly affected by the absence of a host, because it produces much less biomass than when it is growing with the host. Now, a few years back, we did a study, an estimation of how important are these parasitic weeds in rice production in Africa. So we generated these maps and we overlapped. So all the little dots that you see, those are the specimen of herbarium where those parasitic weeds are found. And the green grayish areas is where rain-fed rice is grown. And we estimated based on this that Striga causes $158 million annually to the African economy. And Ramficarpa causes 111 million. But all cereals combined, Striga causes 4.7 billion losses. So also in sorghum and in maize, it is actually very, very important. Now I will give you a short outline of my career to show, to let you know how I ended up with these parasitic weeds. My first experience was, my first African experience was in Burkina Faso, as Andrew already said. That was in 1997, and I was an intern that was part of my MSc program at Wageningen University. I was an intern at a Dutch-funded development project there. And I studied the use of compost pits and compost in sorghum fields by farmers. And I also studied the farmers themselves, surveyed the farmers. And this was also the time when I first encountered Striga. It was also the first and the last time that I wrote an entire project report in French. A year later, I went to Indonesia, completely different experience there. I was working in a field. This is a picture of the field, very steeply sloping field that was slashed and burned of vegetation to grow rubber trees, and in between the rubber trees, upland rice. And to get to this field, you had to cross the river, a river on a raft that you see also on the picture. And on this particular picture was my supervisor, Meijner van Noordwijk of the World Agroforestry Center. And the river was swarmed by blood-sucking leeches, so it was you really wanted to stay on that raft. On the other picture, the right-hand picture, you can see me standing next to the farmer of the field where we were working. And on the below picture, the bottom picture, you can see me in rice. This was also my first experience with rice. And this was my colleague from Brawijaya University in Malang, who was doing his MSc thesis research at the same time as me. And then I really got a taste for research. I really knew that, okay, this is what I wanted to do. I wanted to work in an international organization as a researcher. And I learned that I had to do my PhD. So I was extremely happy when I was appointed as PhD candidate in a group of Martin Kopf at Wageningen University under the supervision of Lammert Bastiaans and Aag van Ast. And I worked on Striga. So this is how I ended up with parasitic weeds. I studied the defense mechanisms against Striga in sorghum. And that work was done in Mali. And these are pictures of some of my other supervisors, Deel Hess and Eva Weltzien. And on the picture below the date, you can see me injecting ethylene gas in the soil together with I had a personal assistant. It was really nice. His name was Anna Dolo. He was great. And we injected ethylene gas to create Striga-free plots in our field. The ethylene gas functions as a germination stimulant of the Striga seeds in the soil. And when there is no crop, when it is bare soil, then the Striga will die in a matter of days. So that is how we created Striga-free plots in a Striga-invested field. And that was essential to also look at how the crop is affected, to look at the relative yield losses. And the relative yield losses, they are depicted here on this figure. On the y-axis, I hope you can read it. On the x-axis are the Striga numbers, that is a measure of infection. And the green dots, that show the genotype, the relative yield losses of a particular genotype. different data points. And I found out that there are also sorghum varieties that are much more resistant, so they have lower levels of infection and therefore also reduced relative yield losses. And there were also genotypes that were tolerant. They did not necessarily have lower infection levels, but they did certainly have lower reduced relative yield losses. So conceptually you can see it as this. The relative yield loss on the y-axis again and infection on the x-axis shows this curve. With resistance you can bring down the infection levels and thereby the relative yield loss. If you have tolerance, you get into another type of curve, so you are also reducing the relative yield losses. And of course, when you combine resistance and tolerance, then you are really in business. You can really avoid terrible yield losses because of striga. After my PhD work, I started at the Africa Rice Center. Again, a completely different experience. I worked under the supervision of Paul Kiepen and also under the directors, the general directors, Kanaya Nuance and Papa Sek, and the director of research, Marco Wopperijs, for part of my tenure at least, and they were really instrumental and supportive of my work. And I resided in different countries together with my family in Africa. The orange ones are the countries where I lived, and the yellow countries are the countries where, in addition, I worked. And it was a great experience, and I was appointed as an inland valley agronomist, and that meant that I had to work in these kind of rice growing environments. They are the inland valleys. And that is sort of a lower lying area, and they are considered to be rain-fed lowland rice growing areas. And in those inland valleys, but also in the uplands, the parts of the field that were more higher up in the upland lowland continuum, I encountered a lot of parasitic weeds. So first in the uplands, my old friend Striga hermontica, but also in the lower parts, a new parasitic weed that I didn't know by that time, and that was Rhamphicappa fistulosa. And here on the picture you can see me in different countries, because all across Africa I encountered these weeds. And I decided to start a study, actually to repeat a study, a survey that was done in Benin about the spread of this parasitic weed in Benin, in different inland valleys. And I revisited the same inland valleys across the country to see whether there were inland valleys that were previously not infested and that were now infested. That was 10 years after the first survey. And I identified indeed that 2 locations where this parasitic weed was new. So it was really spreading, and that got my attention. And I decided to work on this parasitic weed as well. Now, this does not completely answer why I studied them, of course. And to answer that question, I have to show you pictures of the farmers. So all of these farmers, they are holding these parasitic weeds in their hand, different species of parasitic weeds. They're all rice farmers, and everywhere I went for my field work, I encountered those farmers, I talked with them, and they were really troublesome to them. And so for me, it's, from a scientific point of view, a very interesting topic to study. For them, it was serious business. It was really impacting on their yields and on their livelihoods. So that is why I decided, yes, I should work on this. I should try to find solutions, because the farmers were also in the dark. Especially for Rhamphicappa fistulosa, they did not know, it was a newly emerging parasitic weed, they did not know what to do against it. And so I thought, like, this is where I could really make some impact. So my research objective was to find suitable solutions against parasitic weeds in smallholder crops in Africa. And with suitable, I mean, well, there's a range of criteria, actually. They need to be effective, of course, accessible for the farmers that are meant to work with those solutions, affordable for them as well, and feasible. They should be practical. And, of course, also not harmful to the ecosystem health and human health. In order to do that, I had to test the efficacy of control options. I had to learn how control options work. I had to elucidate the ecology and biology of these parasitic weeds because much was still unknown about them, especially Amphicarpa. And I had to understand the host-parasite interactions. So that brings me to the second half of my lecture, and that is, did I find out how to get rid of them? Well, the short answer is no. So if you are now terribly disappointed, you can discreetly leave the meeting room. But if you are interested in the slightly longer answer, and that is that we made good progress, then you can remain seated. So, first I will talk about Striga hermontica and Striga asiatica. Again, I have to start with the life cycle of Striga. For Striga asiatica and Striga hermontica, the life cycle is exactly the same. It always starts with seeds. When the seeds are in the soil and get into contact with the host plants, they can germinate. Then they will form this haustorium to be able to connect to the host root, form a vascular connection, below ground development, above ground development, and that is then followed by flowers and seed production. I'm showing this because there are really 3 different stages in that life cycle that can be targeted from a management perspective, but also from a genetic perspective of the host plant. In the below ground stages, you can target the parasitic weed by pre-attachment resistance. There are crop varieties, genotypes I used to call them, that produce fewer of these Striga germination stimulants. And these are considered to be have post-attachment resistance. They stimulate fewer of the seeds to germinate, and hence you will have lower infection. Another mechanism of resistance is called the post-attachment resistance. So those, and that is the yellow part of this cycle. That is the area where that is the stage where the host plant is able somehow to reduce being infected, getting infected. Not avoid getting infected, but it avoids getting the parasite is not able to make a viable vascular connection with the host plant. And I will explain that a little bit later as well. And then you have, and so both of these mechanisms will bring down the Striga infection levels and hence increase the crop. But you also have tolerance. I already talked a little bit about that, but tolerance is, despite being infected by the host, or let's say reduced, having less impact in spite of the infection levels. So, and that is obtained by the ability to maintain good high levels of photosynthesis. So these plants, they may be affected as much as susceptible plants, but they are much less impacted, and that is called tolerance. In rice, I will flip-flop a little bit between rice and sorghum, if you don't mind. In rice, I started to screen a wide range of popular upland rice varieties against Striga hermontica and Striga asiatica, and I did that in East Africa. So I screened the new rice for Africa, popular new rice varieties, nericas, against local and adapted and preferred varieties and international checks. And I did that in Kenya and Uganda against Striga hermontica, and in Tanzania, southern Tanzania, against Striga asiatica. And this is just one picture actually 2 pictures of the field. At the background you can see Lake Victoria, so this was at the source of Lake Victoria in Kenya. And you see on the foreground you see Nerica 9. I hope you can see it on your left-hand side. And Nerica 9 is completely hammered by Striga. Next to it is Nerica 1, another Nerica variety, and it's doing perfectly fine. So basically what we discovered is that there was a wide range, a wide genetic variation in resistance against striga in rice. And that was also confirmed by another screening trial that we did later on against striga asiatica and hermontica, and that is shown on the graphs below. You don't need to be able to read the details, but you can see the trend. There's quite a lot of variation. On the y-axis is the striga infection level. So there's a wide variation in striga infection levels across these genotypes. Then we wanted to find out what is it based on? How come? Why are some varieties less affected by striga? And that is when we discovered that in a lot of these, or in some of these resistant genotypes for rice varieties, there was pre-attachment resistance. And by pre-attachment resistance, if you remember well, we mean a lowered production of these germination stimulants. So that is shown on this figure. The strigolactones that are germination stimulants, they were characterized in the root exudates of all these different rice varieties, and you could clearly see the same type of variation as in the field. And then we tested these again in pots, and we saw that it indeed confirmed that with high strigolactone production in the root exudates, you had high striga levels. With low strigolactones, you had low infection levels. This was work that was done in collaboration with Harold Bouwmeester and Jamil Muhammad. We also found out that there is post-attachment resistance. Remember that life cycle of striga with the yellow triangle in the bottom. Post-attachment resistance was also there. Here we have studied rice on rhizotrons, and those are trays, and you can grow the roots of the plant on those trays, and you can open them at any time to see how the roots are doing. But more importantly, you can paste pre-germinated striga seeds on those roots, and then monitor its development in the days following. So you already germinated striga, so there's no pre-attachment resistance at play, only post-attachment resistance. And there we found that, for instance, Nerica 7 was highly infected. The green stuff is the striga here. And on the bottom pictures you see a number of rice varieties, for instance Nerica 1 and Nerica 10, and they were very resistant, post-attachment resistance. And we also looked at the tissue level. Well, first of all, at the root level, how does it look like? What is the phenotype of that resistance? And they showed necrosis at the point of attachment. So the roots of the host plant turned dark and actually died off at the point of attachment. And that did impede the striga plants to make a viable connection. But also at the tissue level, I should just say that if you're not familiar with these pictures, the purple cells that you see here, they are the haustorium of the striga, and the blue cells are the host root cells. And they really want to make a connection with the vascular system, where the transport of water and nutrients and such is happening, and that is the blue circle in the middle. So a compatible interaction shown with the purple frame here is shown like this. It's really making sure that the parasite can make a viable connection with the xylem vessels. And an incompatible reaction, the parasite was just not able to get into that vascular system of the host root. It was growing just around it and sometimes coming out at the other end of the root. So we also established that. And this was work done together with Professor Jolie Scholes and Mamadou Sissoko. In a just completed project on sorghum, we were interested in studying post-attachment events, and we were interested in studying whether we can also increase the resistance and also tolerance by applying fertilizers to those varieties that are resistant or tolerant. Can we enhance it? Because it is never complete. Striga-resistant varieties are still infected by Striga. Striga-tolerant varieties still have some yield losses. So what was already known before that was that crops that are fertilized by nitrogen and phosphorus and P fertilizers, they have less Striga because the host plant is producing less of the strigolactones. So that is a mechanism I will not go in detail, but you need to know that that was already sort of known. So that is why we decided let's study the post-attachment phases. Can fertilizers also improve post-attachment resistance? And can fertilizers also improve the tolerance of the plant? Or in other words, can damage to host plants be avoided by fertilizer application? This was done with our PhD student who completed 2 months ago, Immaculate Mwangangi and Stephen Runo, and also with Lucie Buchy. And again, we studied this in rhizotron experiments because remember, in the rhizotron you can open the tray and paste those germinated Striga seeds onto the host roots. So that is when you want to look at post-attachment resistance. And we tested 5 different genotypes and 4 different levels of fertilizers. This is getting a little bit technical, but I will try to not go in too much detail. We had a susceptible genotype called Ochuti, and we had 4 different genotypes, varieties of sorghum, that had different levels, different mechanisms of post-attachment resistance. And we studied them against different fertilizer treatments. 25%, the base, it's what we called it, base level fertilizer, 25% of the recommended or the optimal levels of nutrients. Micro, that is the treatment that only received 100% of the micronutrients, but still 25% of the macronutrients. And macro is the flip side of that, 100% of macronutrients and 25% micro. And then macro, that was the complete fertilizer application. So all you should remember is actually from base to macro is increasing improved fertilizer application. And this is again a picture of the rhizotron where we can study the roots. And indeed, by applying fertilizers to this system, this rhizotron system, the Striga infection levels on sorghum were really much reduced. On the y-axis again is the Striga infection. And on the x-axis are the different levels of fertilizers. So that was confirmed, and it was actually the first time that it was shown that fertilizers, plant nutrients, also have an effect on the post-attachment resistance and not just on the pre-attachment resistance. Quite novel. And then we looked at tolerance, and tolerance was studied in pot experiments in the greenhouse here at the campus. And we studied them. We studied sorghum, different sorghum varieties. Striga sensitive CK60B in blue, Striga tolerant Ochuti in pink, and Striga tolerant Timariving in green. And we had Striga infested and Striga free pots, so we could really look at the relative yield loss. And again, the same fertilizer treatments, so I don't need to explain that to you again. On the left-hand figure, with the yellow frame, you can see the plants growing without Striga. And you can see, I hope you can see that, a little grayish bar. On the y-axis is the photosynthesis level of the plant that we measured. And all the plants growing in Striga-free conditions, no matter what the Striga genotype was, no matter actually what the fertilizer levels was, which was a little bit surprising, it all stayed within that band of 30 to 40 micromole per square meter per second. That is the expression of photosynthesis. But when those plants were Striga infected, and that is with the red frame, under the same treatments, you could really see that the blue variety, the blue genotype, is the sensitive one, is really dropping their photosynthesis levels because of the Striga infection. But improved fertilizers increased that photosynthesis level of all the varieties, but the tolerant ones were brought back into that gray band area of 30 to 40 micromole per square meter per second of photosynthesis. The sensitive one, Striga or CK60b, was not able to get back to those levels. So the answer to the question, can the damage of the Striga be avoided by fertilizers, is yes, but only when you already have some tolerance in the background of these genotypes. When they are sensitive, there's no way you can recover from the losses. And this is also depicted in these pictures, the photos. You can see from Striga free to Striga infected, Striga free to Striga infected plants. You can see that the sensitive genotype is able to do a little bit better with improved nutrition, but you can see the panicles, where the grains are, is really not getting back to the same level as the Striga free plants. But on the Striga tolerant sorghum genotype, you can see that it is completely recovered. So that was also a very nice result. And then I'm going back to rice again. I told you that I will be flip-flopping a little bit. And this work was done in Madagascar. Very shortly, briefly, but I wanted to show that we have also done work on agroecological practices, also to see whether or not we can strengthen the Striga resistance in rice by applying management practices. After all, I'm an agronomist. So this was done in Madagascar in an area that was infested by Striga asiatica. And on this picture, you can see the Striga infection level again on the y-axis, and three different rice varieties on the x-axis. The first one is B22, that is popular among farmers there, and you see that they have high infection levels. But when you replace that variety by a resistant rice variety, it has lower infection levels. Well, we already established that. But when you apply these agroecological practices, and those were actually preceding the rice by a maize legume intercrop crop. So in the year before rice, growing maize with a legume intercrop, then you also reduce the Striga even further. And I will not go into detail, but we tested this with different legume crops, and you could also see that there were differences in the legume crops in helping the Striga, bringing down the Striga, and enforcing the already good resistance that is there in the varieties. And this was done with a PhD student, Tahir Randria Javisanaka, and Alain Paul Andrea Naivo, and Patrice Autfray of Sirat. Now I will talk about Rhamphiocarpa fistulosa, because this was, when I started working on that, was a completely new parasitic weed. There was not much known on that before. So it was really pioneering work and really exciting actually, because there were lots of things that I needed to find out. The first thing, one of the first things that we did was looking at the life cycle of this parasitic weed, because we knew it was different from Striga, of course, because it is a facultative parasite. So we established that, and the germination and the emergence of this parasitic plant takes about six days and does not require host root exudates. It does not require germination stimulants from the host. It can perfectly germinate with water and light. All it needed is water and light to germinate. That is very important in terms of the management. For instance, the legumes that I showed you before that are actually false host crops, and they stimulate the germination of the Striga, but they cannot host the Striga, so the Striga will die. That type of innovation cannot be used against Rhamphiocarpa fistulosa. The maximum germination and actually the start of the parasitism is 36 days later, so after sowing, so much later than with Striga, and with Striga it is immediate. And then it really starts to gain biomass from parasitism. It flowers, it matures seeds, and then the rice plant, the yellow and brown circle, the rice plant starts to sense and really get affected by the parasite by increased root-shoot ratio about a week later after parasitism. And then it gets reduced light interception, arrested biomass production, reductions in photosynthesis, tiller numbers, leaf area, etc. So that means that we have actually a window of control for Rhamphiocarpa. Because if you can control Rhamphiocarpa before it attaches to the host, you will not have any problems. And that window of control is about 36 days. So that is quite important. You don't have that window of control with the obligate hemiparasite Striga because it germinates and attaches below ground and is infecting it straight from the start. We also identified another window of control, and that is because the Rhamphiocarpa itself is flowering and producing seed much earlier than the completion of the rice crop. And so if you are able in that 50-day or so period to remove the Rhamphiocarpa, which is a lot of work of course, but then at least you can avoid that the seeds seeds are produced and causing problems in the next season. Now we did a screening of the rice varieties, but different rice varieties, but with the same name actually, Nericas, but those Nerica varieties, they were adapted to the lowlands. And the rain-fed lowlands, that is where Rhamphiocarpa was really a problem. And so that was a wide range of varieties again, and we screened them, and on this picture you can see that the green part, that is the rice. All that is not green is not good, that is the parasite. So there was also here, we found a lot of genetic variation in Rhamphiocarpa resistance. And that is also depicted on in this figure. The blue points are these Rhamphiocarpa infection levels. On the y-axis, you can see the Rhamphiocarpa infection, the biomass, and on the x-axis you can see the rice varieties. Again, you don't need to see the details. It's just the principle that we were able to identify, which was quite exciting. We didn't know that before, that there are actually some rice varieties that are quite resistant against Rhamphiocarpa. And then we also identified a wide range in the relative yield losses. So not all rice varieties were equally affected by Rhamphiocarpa. Again, a very nice find. So we identified also some tolerant rice varieties. And then we also wanted to see, okay, can we do something with fertilizers? Because fertilizers proved out to be very effective against Striga, right? So we started to look at how do fertilizers affect this facultative parasitic weed, and how does it impact on the crop. So we studied the host alone, and then the parasite alone, and then the host and the parasite growing together, with 6 different fertilizer levels, from 0 to 200% of the optimal rates. Here you can see nothing earth-shattering. This is a host plant rice fertilized by improved levels of nutrition, and you can of course see a very clear increase in the rice biomass, nothing new there. But when it is infected by Rhamphiocarpa, then you see that the biomass is steeply reduced, and you see that the slope of this line is also reduced. So it is actually not benefiting from the fertilizer when the parasite is there. The host, on the other hand, when the parasite, when Rhamphiocarpa is growing all by itself, no matter how much fertilizer you apply to it, it does not respond to the fertilizer. It is a flat line. So that is also interesting. No response of the parasitic plants to fertilizers. But when it is grown with a host, then all of a sudden it can really benefit from fertilizers when it is infecting the host plant. And now something interesting also. The red line is the Rhamphiocarpa biomass grown with a host. The blue line is the rice, the host with the parasite, the biomass. When you add those two biomasses up, the total is still always lower than the rice plant growing alone. So that is a phytopathological effect that is shown here. So it's not just a sink-source relation, like what is gained by the parasite is lost by the host. It's much more than that. There's much more happening there. And these are just some pictures of this experiment, so you can see for yourself. No fertilizer on the left top, and 200% or 100% of fertilizer underneath it. You can really see that the Rhamphiocarpa is not benefiting from it, and the rice is also not producing any panicles when it is infested by Rhamphiocarpa. Now I'm almost there. Then we really wanted to study these parasitic weeds in the field, and we were lucky because we found a place in southern Tanzania, in Kyela, where both Rhamphiocarpa and Striga asiatica, were grown in the same landscape. And so they were sometimes affecting the same farmers that had fields in the uplands and in the lowlands, sometimes different farmers, but at least very similar environmental conditions, climatic conditions, and this enabled us, with the same farmer groups, to study those parasitic weeds conveniently in one area. And this aerial picture is showing that Striga asiatica in the uplands and Rhamphiocarpa fistulosa in the lowlands. And on this picture is also, I also show Dennis Tippe, who was one of our PhD students, and Stella Kabiri in red. And she is standing next to Lambert Bastiaans and Aad van Asten. Maybe you remember those names. They were actually my old PhD supervisors, and we started a new project together on these parasitic weeds in rice years later. So we did this work also with farmers. So we studied Striga asiatica in the uplands and Rhamphiocarpa in the lowlands, and these are the results of when we apply fertilizer. So there's the no fertilizer control on the left, then there is mineral fertilizers, NPK and DAP, and organic fertilizers. And you see a little bit the same trend as what we have seen in the pots, that the Striga asiatica is nicely reduced by applying fertilizers, whether mineral or organic. The Rhamphiocarpa, on the other hand, is showing a little bit the response that we also observed in the pot experiment. Rhamphiocarpa is, with improved fertilizers, is actually gaining. But surprisingly, and also disappointingly a little bit, was that it was gaining much more from organic fertilizers than from mineral fertilizers. We also did a sowing time experiment to see, does it matter at what time we establish the crop? And so these, the sowing times were altered by 14 days, so each time 2 weeks later sowing. And the delayed sowing really reduced the Striga infection levels, but it increased the Rhamphiocarpa infection levels. And that's because Striga seeds need to precondition in the soil, and that is by moisture, soil moisture. After a while, when they cannot germinate, they will go back into dormancy again. So that explains that when you delay it, you will have less Striga that can germinate. And the Rhamphiocarpa is a very low starter, slow starter. So if you sow earlier, you sort of expand that, you enhance that sort of time advantage that you have there. And like I said, this was also done with farmers, and so they were completely involved in these experiments. It was also done on their farms and their management. And then we asked them what would be feasible and interesting innovations for you. And so the farmers in the uplands dealing with Striga asiatica, they said, well, indeed later sowing would be good, but then we need shorter duration varieties. And we did all of this work also with different varieties, and they chose the short duration variety Nerica 10, which was also Striga resistant. And the organic fertilizers, they saw that as quite a good alternative for the mineral fertilizers, which cost much less. So that was quite a nice result. In the lowlands, early sowing was indeed preferred because of the reduced Rhamphiocarpa infection levels, and it was combined with long duration Super India. So that is their preferred varieties. So because they established the rice early, they could hold on to their preferred long duration variety. They had mixed feelings about about the fertilizers, about the organic fertilizers, because, well, it did increase the Rhamphiocarpa, of course, but it also improved their rice yields. So there was no consensus there. This brings me to the conclusions and the acknowledgements, and I'm sure that you are happy because I can be a little bit long-winding. Parasitic weeds have a very strong impact on crop production in Africa. I think we have established that. But also on careers of researchers studying them, because I did my PhD work on that years ago, and here I'm standing more than 20 years later, and I'm still working on these same parasitic weeds. A few things can be done to reduce the infection and alleviate post-infection damage of these parasitic weeds. But I have established, and I hope that it has also become clear from this lecture, that the biology and the ecology of the parasitic weeds really determine how best to manage these weeds. But varieties with increased levels of resistance and tolerance, they are really the backbone of any integrated weed management approach against them. So it should all start with good material, good resistant or tolerant varieties. Against striga, it can be combined with fertilizers and legume companion crops, or delayed sowing. And I put fertilizers and the legumes here in red because they cannot be used against Rhamphiocarpa. In Rhamphiocarpa, you can combine the resistant and tolerant varieties with early sowing and pre-attachment removal. And here pre-attachment removal is in red because it cannot be done against a striga because the attachment takes place below ground. I brought my book with me. I published a book just a month ago, but I just realized it is somewhere in my bag there. But I published this book together with Professor Lytton Musselman on parasitic weeds in parasitic plants in African agriculture. And we are treating all the 47 parasitic weeds in African agriculture that are known, and also some that are really not known, and we describe them. And I would like to thank the University of Greenwich, the Natural Resources Institute, and all the collaborating organizations I worked with through the years. And on the bottom of this picture are my main funders, which I would also like to thank. And then I come to thanking people, and with that one I really struggled. First of all, I listed down all the people I worked with on my slide, and it became really crowded, and then I was worried that I would forget people. So I think the best and safest way is just to say thank you all. Staff and students of the crop and weed ecology and the plant production systems groups at Wageningen University. Thank you all to the staff and students of the Africa Rice Center, staff and students of the Natural Resources Institute, and I'm happy to see that there are even some of my master students who I supervised last year are here with us. And I would like to thank my family in the Netherlands and also my family here in the UK, of course, for keeping up with me, for coping with me. And I would like to thank Caroline Troy for organizing this. And Lucie Buchy, because she's not here, but she's my close colleague, and she helped me in preparing this lecture. Thank you very much.
I think we've worked out why Jonne gets up in the morning. His passion for parasitic weeds drives him up and into the office. So I'm Cheryl Hendricks, I'm the director of the Natural Resources Institute, and we're very proud of the work that you've done and your investment in improving the lives of people in Africa. So we can see that you're driven by passion from the people that you've shown us today and how you've engaged with the struggles on the daily struggles on the farm and how that drives what you do. But yeah, who would have known you could make a career out of weeds? So that's really fantastic, and I think your ability to tell a story as a top scientist, to be able to convey quite complex science in a way that is understandable to many people is a real gift. And thank you for giving us a window of insight into your world today. I was struck by the humbleness by which you never told us that you were a pioneer and had named a plant, although it not be a Latin name, but that you had named a plant. So that's really wonderful. I was struck really by the amount of food that's lost. So that was something that wasn't on my radar. We have many scientists at NRI who study post-harvest losses, for example, but your number, which I wrote down, $4.7 billion lost due to weeds, the total amount of food traded in a year in the world is 12 billion. So that's about a quarter of the total traded value that's lost before it even gets to be harvested. So something that really should perhaps be made more publicly known, that it's not just from post-harvest loss, but before as well. So thank you so much for giving us a window into your world as what you do every day and what drives that passion. And hopefully everybody has learnt something from it. We won't be handing out exam papers. Instead, we'll be handing out snacks and drinks. So in tradition of a public lecture, there are no questions at the end, so you can grill Jonne over a drink outside in the foyer area here. But just to thank your family for being here and for the support. We know that they support you daily. So thank you. Just a thanks to Andrew, our DBC, Peter, our PBC, and Richard, Jonne's head of department, for supporting and being here today. And Ben, Deputy Director of NRI, thank you for being here. It's been a long day for Ben. So yeah, and thank you very much, Caroline and Josh, for the support and backup in presenting this. So can we give Jonne another big round of applause?